My name is Rachel Stander, and today I will be presenting on the effects of road salt and its alternatives on freshwater invertebrates. To begin, road salt is an incredibly common and effective tool used to melt ice and snow on the roadways during winter months. It is primarily composed of sodium chloride, or NaCl, and it works by lowering the freezing temperature of water, which then results in a quicker melting time. It is an inexpensive and easily accessible de-icing agent used by many municipalities. And something that I found really interesting is that the quantity administered of road salt has increased tenfold in the last half century. That meaning that in 1950, there was about 1 to 2 million tons of road salt administered. And today, there is on average about 10 to 20 million tons of road salt administered onto our roadways. And with such an increasing amount of road salt being used, it leads me to question what are the implications of road salt administration. And to begin, water toxicity of freshwater systems, which includes sewers, rivers, groundwater, and lakes. And then also for the focus of this presentation and the focus of the study that I ran, salt pollution harms aquatic plants and animals. It also leads to salt corrosion on cars and affects groundwater quality and contaminates drinking water as well. And then lastly, road salt leads to increased vulnerability of exploitation by invasive species. So, and there's many more that I could list for you, but these are just some of the major ones, and just to outline that there are many implications of administering road salt. So to move on to the experimental objectives of my study, we aimed to examine commercially available road salt alternatives' effects on invertebrate species. And how we examined these was by measuring the survival and reproduction of several invertebrate species. And so the hypothesis was we believe the alternatives would affect the invertebrates differently, and some would be more extreme than others. So the alternatives to road salt that we selected for this study included a beet juice ice melt, sugarcane molasses, pickle juice or brine, and then sand. And so to go over how these are effective at melting ice and snow, the beet juice contains sugars which actually melt, which actually lower the melting point of ice, and it is also fully biodegradable. For the molasses, similar to the beet melt, it contains sugars that lower the melting point of ice. And then for the pickle prevents ice from bonding to the pavement and reduces the amount of chloride ions in the environment. And then lastly, for sand, as we know, sand doesn't actually melt ice, but it is an abrasive material that provides traction on the roads. And then for our study organisms, we selected Daphnia pulex, Ostrocodia, midge larvae, and then a species of earthworm. And the pictures of those 4 invertebrates are at the bottom of the slide. And then lastly, it's important to note that the ostracods and the midge larvae were collected from the Maple River Salt Marsh, which is the only intact salt marsh in Michigan. And it's interesting because these invertebrates already exist in an environment with natural salt deposits. So we thought that would be interesting to look at in comparison to invertebrates that exist in a freshwater-only environment. So the experimental procedure and how we expose the invertebrates to the treatment is outlined here. We have at the top on the left side, we have the 4 treatments, alternatives to road salt, which is the sand, molasses, beet melt, and pickle juice. And then we also tested road salt as a treatment just to see how they would react, and then as well as a control of spring water. So for the Daphnia, the ostracods, and the midge larvae, they were put into small glass cups with a concentration of 2.5 grams per liter of the treatment. And then on the right picture, you see these plastic deli containers filled with soil, and those were for the worms. And of course, worms, they live in soil, so we had to make an accommodation for them. So instead of being completely submerged in the treatment, they were sprayed daily. And you can see the spray bottles on the right-hand side of that image. We sprayed each deli container with the worms in it every single day with the same concentration that the other invertebrates were exposed to. And then continuing with the methods, we collected data on survival and reproduction daily, as well as changed the water once a week. So the water was not stale that they were living in throughout the experiment, as well as feeding all the invertebrates once a day. And then what we decided to do was calculate the integrated population density for each of the organisms. And we used the trapezoid rule to calculate the area under the population growth trajectory. And this method allowed us to accommodate the fact that we did not collect data every single day. And it also allowed us to account for asynchronous population changes across replicates, especially the fact that both replicates increased and decreased, population increased and decreased in all the replicates throughout the experiment. So moving on to our results, to start off with the Daphnia, we found that the Daphnia were strongly impacted by the pickle juice and the road salts, and they were intermediately affected by the beet salt and the molasses. So the two graphs that you see before you, on the left we have a box plot, and on the right we have a line graph, and they just, on the box plot, it's represented by IPD, the integrated population density, and then for the line graph on the right, it outlines how the population increases and decreases throughout the experiment. And based on an ANOVA that we ran on the data, we found significant differences between the salt and water, the pickle and sand, the pickle and water, and the salt and sand for the Daphnia. And moving on to the ostracods, we found that the ostracods were only impacted by the pickle juice. And so as you can see on the box plot on the left, as well as the line graph on the right, the pickle juice treatment, being the green one, is far significantly decreased for the population size of the ostracods as opposed to the other alternatives. So again, after running an ANOVA, we discovered that there was a significant difference between the pickle treatment compared to all the other alternatives for the ostracods. And then lastly, for the worms, we found that the worms were not impacted by any of the treatments. After running the ANOVA, we found that there was no significant difference in integrated population density among the different alternatives for the worms. And something important to note for the worms is that the y-axis on this graph does not go down to 0. So overall, the worms population stayed about the same throughout the whole experiment in all of the alternatives. And then now for some conclusions and take-home message. One note I would like to make just to clarify is that the midge larvae were not included in the results graphs, and that is because the sample size of the midge larvae was very small and it was too small for statistical analysis. So for our conclusions, the first one is that invertebrates perform better in different alternatives, and specifically beet, molasses, and sand are typically the less harmful alternatives for the invertebrates. We found that the ostracods, which come from, of course, a naturally salty environment, the ostracods were collected from the salt marsh in Michigan, which have natural salt deposits. And then the worms, which are, again, they live in soil, and that correlates to being far less exposed to the treatments as opposed to the other invertebrates. And so the ostracods and the worms were less affected by the salt than the other invertebrates. And what we have found is that high concentrations of road salts are found to be detrimental to the population growth of subinvertebrate species. And so for our future research, we hope to continue searching for deicers that are non-toxic to freshwater ecosystems and invertebrates. And then lastly, the take-home message for this project is that we want to try to limit the amount of road salt that is exposed to the environment because it all collectively is detrimental to invertebrate species, aquatic invertebrate species, as well as terrestrial invertebrate species. And just a couple of acknowledgements I would like to make. I would like to thank Kurt Hellman, Chris Breen, the Albion College Biology Department, and then the Foundation for Undergraduate Research, Scholarship, and Creative Activity, or FIRSCA. And that includes Dr. Vanessa McCaffrey and Renee Krieger, who run FIRSCA, and thank you to them for funding this project. And that is it. Thank you.